Cell junctions play important roles in the functionality of our tissues, including stabilizing the tissue structure, as well as coordinating the cells functionally and directing those cellular processes, ensuring that the cells individually and as a tissue respond appropriately to changes in their surrounding environment. Cell junctions are grouped into three categories: those that anchor cells to each other or to extracellular structures, tight junctions, which are unique to epithelial cells, or communicating junctions. Most of the material in this lecture comes from a book called The Molecular Biology of the Cell, which you can access to learn more about these topics from the NCBI bookshelf. We'll start with anchoring junctions. These are present in most tissues throughout the body, and they're particularly abundant in tissues that are subjected to routine mechanical stresses, such as the gut, lungs, tissues that are constantly undergoing movement. We can divide them into two classes based on their transmembrane adhesion proteins. Adherens junctions and desmosomes connect cells to the neighboring cells, and they utilize the cadherin family of proteins. In contrast, the junctions that bind cells to the extracellular matrix, focal adhesions and hemidesmosomes, utilize integrin proteins for these attachments. Cadherin proteins depend on the presence of calcium to bind to one another. This is a trait that's been made use of in many laboratories where they grow cells in vitro in order to passage the cells to break those bonds. One simply removes the calcium and divalent ions from the surrounding medium, and this effectively unzips all the cadherin proteins and allows the cells to be separated from each other without damage to the actual proteins. In humans, there are over 100 different types of cadherin proteins, which are generated both by different types of genes, as well as alternative RNA splicing of those genes. The result is a whole family of very specialized cadherins, depending on the tissue, the location, and the surrounding microenvironment. Loss of epithelial cadherins can play a role in oral squamous cell cancer, as those cells then become less able to adhere to their neighboring cells. Without that epithelial cadherin, they're more likely to break off, and this leads to invasion and metastasis of the cancer. And for this reason, researchers are looking into agents that restore functional cadherin as a way to treat these types of cancer. There are two types of anchoring junctions that utilize cadherins. They're the adherens junctions and the desmosomes. Adherens junctions connect cells together and can affect the three-dimensional arrangement of sheets of epithelial cells. Between cells, cadherin dimers bind to each other within that intracellular space. The cadherin proteins themselves cross the cell membranes and inside the cell bind to proteins which link them firmly to the actin filaments within the cells. Groups of adherens junctions linked to these actin filaments then can cause something called an adhesion belt. And in this way, adherens junctions are linked to a beltwork of active filaments that extends throughout the epithelial tissue, and furthermore is able to tighten or loosen based on cellular signals. And this is important in determining our three-dimensional tissue structure. An example of this is the formation of tube structures during embryogenesis. Initially, you have a sheet of epithelial cells knit together by adherens junctions. Within this sheet, some of the adherens junctions begin to tighten, whereas others loosen. This is similar to pulling a thread in a piece of material. a U shape begins to form, and eventually a fully formed epithelial tube pinches off from the overlying sheet of cells. The formation of adhesion belts throughout an epithelial sheet is what induces epithelial cell contact inhibition. This is a signal to epithelial cells to stop dividing and instead to begin the process of differentiation. And loss of this signal is a hallmark of cancer cells. Adherence junctions must also be present and intact before tight junctions can form, an important step in the differentiation of epithelial tissues. Desmosomes are another type of anchoring junctions that utilize cadherin proteins. Again, between the cells, cadherins bind to each other. The cadherin proteins have a structure that crosses a cellular membrane, and inside the cell, they bind to what's called a protein plaque, which is made up of many different types of intracellular proteins, anchoring proteins. And you can see these plaques clearly in this electron micrograph showing desmosomes connecting two cells together. They're like button structures or rivets that join these cells strongly together. And the cadherin proteins are linked then, bound via these plaques to the actual cytoskeleton of the neighboring cells, so that the strongest structural parts of the cells are bound together by the desmosomes. In this beautiful electron micrograph of a desmosome, you can see how extensively the intracellular plaque is bound to the filaments of the cytoskeleton. Desmosomes contribute to the structure. They hold the cells together, but they also give great tensile strength to the tissue as a whole because of this extensive connection to the cytoskeleton. There are specialized cadherin molecules that are only present in desmosomes, and they fall into the families of the desmoglines and the desmocollins. Between cells, again, these proteins form dimerize to hold the cells together. Within the cytoplasm of the cell, they are bound through a series of connections to placoglobin, desmoplackins, and then to the intermediate filaments. The importance of desmosomes to structural integrity of the organs is shown clearly in some conditions where the desmosomes are non-functional. Pemphigus vulgaris is a disease in which there are antibodies that attack a person's own cadherin proteins, and specifically the cadherins present within desmosomes. Affected individuals have blisters, painful blisters that are slow to heal in the gingival surface and the buccal surfaces within the mouth. You can also have, in severe cases, severe blistering of the skin with leakage of body fluids, and these severe cases can be fatal in 5 to 15% of affected individuals. Carvajal and Knoxville syndromes are similar in that they also involve non-functional desmosomes. In this case, the affected individual has a mutation in one of the desmoplakin genes. An interesting aspect of this disease is that it impacts tooth formation and in fact prevents the formation of the adult teeth, so that affected adults then have a mixture of adult teeth and baby teeth that they never lost. And as a dentist, when you see this characteristic, particularly in combination with a very heavily, very thick cornified surface of the hands and a texture of hair that's often very thick and woolly, these symptoms of Carvajal and Knoxville syndromes indicate that there could also be cardiomyopathy in individuals with these syndromes, where some of the heart muscles are replaced with non-muscle tissue, and it can result in sudden death. So as a healthcare provider, if you were to see these qualities in a person, you might then be able to recommend that they have their heart checked. Anchoring junctions that are formed with integrin proteins include hemidesmosomes and focal adhesions. Integrin proteins function in multiple ways. They both hold cells together and they contribute to signaling processes between the cells and the extracellular matrix. Integrins operate on a Velcro principle in that there are many integrin molecules present throughout tissues. Each of them has a low affinity binding, but because there are so many of them, they can result in very stable bonds, and yet these bonds are easily changeable as the cells respond to changes in their local environment. So integrin-mediated adhesive interactions are important for cell migration and proliferation, the process of differentiation of epithelial tissues, as well as signal transduction. Hemidesmosomes connect cells to the extracellular matrix around them. Within the cells, there is a cytoplasmic plaque composed of multiple proteins, which is then anchored firmly to the keratin filaments, which are part of that strong cytoskeleton. Outside the cell, the integrin proteins bind to multiple molecules within the extracellular matrix, including collagens and different types of laminin molecules within this tissue. And the binding of these proteins creates long fibrils that can extend deep into the extracellular matrix. This is particularly crucial in the skin, where it is the strength and numbers of these fibrils that allow the dermis and the epidermis to be so tightly bound together. Some of the proteins in the plaque of hemidesmosomes come from the same families as those found in desmosomes, so plaquins. And so it is unsurprising that when there are defects in these proteins that affect hemidesmosomes, they can also result in blistering diseases, similar to what we see with in patients with non-functional desmosomes. Hemidesmosomes are also important in the junctional epithelia. This is how the gingival tissue within the mouth binds to the enamel surface of the teeth. In periodontal disease, one of the hallmarks is that the gingival epithelia does not adhere to the tooth surface. And this is partly due to damaged or missing hemidesmosomes within these cells. Here you can see in a healthy gum, the gingiva is tightly bound to the enamel surface of the tooth, and so there's very little space between the two tissues. In contrast, in periodontal disease, this pocket of space between the gingiva and the tooth is much deeper and more extensive. And it's been found that in chronic periodontitis, some of the integrins that form the hemidesmosomes are simply missing in these tissues. And in addition, there are also loss or changes in the abundance of the collagens and laminins, these tissues that the fibrils bind to, forming that strong connection of the hemidesmosome to the extracellular matrix. There are similar changes that are found in other types of periodontitis as well. Focal adhesions also bind cells to the ECM. And in addition, they allow the cells to respond to stresses or changes in their microenvironments. Outside the cell, the integrins are bound to the ECM through multiple bonds with collagens or laminins. Within the cell, the integrins bind to a plaque that includes proteins in the actin talon or filament families, and vinculins, and ultimately to actin filaments within the cell. In this fluorescence micrograph, the actin filaments are shown in green, and the vinculin is stained with a red fluorescent marker. This is a cell in a petri dish, a fibroblast, which is in the process of moving. It utilizes focal adhesions that are formed at the leading edge of the cell. When the focal adhesion is bound to the surface, the filaments then retract, which pulls the cells forward. The focal adhesions can then be disassembled and reassembled in another location in order to change the shape of the cell or permit the cell to move within its microenvironment. Focal adhesions are dynamic. They can form, change, disappear in response to many different signals, either coming from the cell itself or received from the external environment. Focal adhesions can transmit mechanical signals from the tissue itself to the cell via these integrin receptors, and the receptors in turn are bound to many different types of proteins within the cells. So focal adhesions can sense changes in the environment and cause the cell to respond in many different ways. Proteins, GPases, kinases, phosphatases, many different processes can be initiated as in response to changes in focal adhesions. Non-functional or dysfunctional adhesion proteins can be involved in tumor development, cancer metastasis, again, because of the loss of the adhesion and the loss of the control of cell shape and cell mobility. The process of orthodontic tooth movement is highly complex, but focal adhesions play a role in this process. And I like this image of the focal adhesion because I think it illustrates the amazing complexity and the connections between the focal adhesions and so many of the different cellular processes. During orthodontic treatment, the mechanical pressure on the soft tissues creates a constant stress that is detected by cells via the focal adhesions. And the cells respond to this stress in part by removing the focal adhesions that are under the most stress and then forming new adhesions that are in a less stressful conformation. And the result in this are changes in cell shape and positioning that lead to remodeling of the soft tissues within the gums. Now we'll move on to tight junctions. These are found only in epithelial cells, and their function is to severely limit and control the diffusion of solutes between the outside and the inside of a tissue. They enable the polarization of the apical and basolateral membranes, which allows proteins in those different membranes to control the permeability to different solutes. Tight junctions are what enable the transport of solutes against a concentration gradient. And this example is from the gut. The apical membrane of the gut epithelial cell is rich in active transporters that bring glucose from the lumen of the gut into the cell against its concentration gradient. The basolateral section of the cell contains passive glucose carriers that allows the glucose to diffuse out of the cell and into the interstitial space where it eventually reaches the blood. Tight junctions prevent the energy-driven glucose transporter or the carrier protein from moving from one area of the cell to the other. And in addition, they prevent any back leak of glucose between the extracellular fluid from leaking back into the gut. Tight junctions are made up of sealing strands of proteins which connect cells together and are anchored in the cytoskeleton of the neighboring cells. So these are cell membranes of two neighboring cells. The sealing strands are buried within the cell structure and then cross the cell membranes. And these strands of proteins are made up of proteins primarily in the families of the claudins and the occludins. These are very specific proteins that are only found in sealing strands. How tightly these strands seal and what their permeability characteristics are determined not only by the number of sealing strands, which can vary from 1 up to 7, but also by the identity of the individual claudins and occludins that make up the sealing strands. In this image, you can see the tight junctions between epithelial cells. Tight junction permeability can change during tissue inflammation, in which the opening of the tight junctions allows the release of tissue fluids so that the cells are not injured by the buildup of excess fluid during the inflammation and swelling. There's also an influx of inflammatory cells, and in this way, the opening and control of tight junction permeability assists in the resolution of tissue inflammation. The last group of cell junctions we're going to talk about are the gap junctions. In contrast to the other junctions, these do not structurally hold cells together. Instead, they're a mode of communication between neighboring cells. They're named gap junctions because many years ago, when scientists were looking at cells through microscopes, they noticed that the cell membranes of neighboring cells were never right on top of each other, but were always separated by this gap of 2 to 4 nanometers. Gap junctions are hexamers present in the cell membranes of each cell, and when they join together between two cells, they form a hydrophilic channel that allows molecules or electrical signals to pass from one cell to the next. They can open and close, and their state of being open or closed is regulated by the cells themselves. Gap junctions are composed of proteins in the family called connexins. There are many different types of connexin proteins, and they can join together in hexamers of identical connexins or of different combinations of connexin proteins. Additionally, the hexamers, which are known as connexons, join together between two cells to form the passage from one cell to the next, and the connexons that join together to create a fully functional gap junction can either be identical or can be different. And these diversity of connexin proteins and the ways that they combine allows for a very highly specialized gap junction that's specific to given tissues or even cell types within a tissue. The permeability of gap junctions, whether they're open or closed, is regulated by a number of different signals from within the cell, such as changes in cytosolic pH or free calcium levels, or extracellular signals such as hormones or electrical signals. A nice example of this is shown below. This is an image of the retinal tissue in which the neurons have been injected by with a fluorescent dye. On the left, Under normal resting conditions, gap junctions between neurons are open. This is what allows the neuron electrical potential to move so quickly throughout an entire tissue. And so when one neuron is injected with a fluorescent dye, it quickly diffuses throughout the entire neural network in the tissue. On the right, the same tissue has been treated with dopamine, which functions to close gap junctions in the retinal tissue, and as a result, we now only see the single neuron that was injected. Gap junctions coordinate tissue functions in several different ways. They allow electrical coupling between cells by allowing action potentials to be transmitted very quickly throughout a tissue. This is important for the heart muscles and smooth muscles of the GI tract, where the movement of the action potential needs to be fast and coordinated throughout the entire organ. Gap junctions also allow metabolic coupling because they allow small nutrients to be shared between neighboring cells. Gap junctions also facilitate signal transmission by neurotransmitters or other actual transmitting molecules. An example of this is in hepatocytes. When they are stimulated by the sympathetic nervous system, they convert glycogen to glucose and release the glucose. However, because not all the hepatocytes are directly innervated, that norepinephrine signal is spread throughout the gap junctions. Gap junctions can also play an important part in early embryogenesis, where closing of specific gap junctions allows groups of cells to differentiate independently. And finally, gap junctions are important in enamel formation during the process of tooth development. This diagram summarizes the types of cell junctions that we've discussed here. The adherence junctions that form these belts that join epithelial cells together and allow them to shape three-dimensionally the tissues. They also permit the formation of tight junctions in epithelia, which control solute movement across the epithelial barrier. We have the desmosomes that rivet the neighboring cells together, the gap junctions that allow communication between these cells to facilitate the function of whole organs, and finally the hemidesmosomes that bind the cells to the extracellular matrix, focal contacts which not only bind them but do so in a very dynamic manner, allowing the cells to respond to changes in their microenvironments.